In this lecture, we'll be looking at the genus Bdellovibrio under the family Bdellovibrinaceae, order Bdellovibrinales, and class Oligoflexia. This used to be in the class Delta Proteobacteria, but now they have moved to a different class, Oligoflexia. So this bacteria is special because it is a bacteria that attacks other bacteria. The type species is Bdellovibrio bacteriovorus. First described in 1963, it is gram-negative and an obligate aerobic bacteria. There are now 4 species found in this genus, and they are mostly found in river water and soil. They parasitize other gram-negative bacteria by entering their periplasma and feed off their host. So there are 2 lifestyles, the highly motile attack phase and the slow-growing Bdelloplast phase. After the attack phase, once they enter the prey, they will form a Bdelloplast and then will slowly convert the host cell into spheroblast. So in the attack phase, they swim very fast, 160 micron per second, and will collide with the prey. Once upon collision, they will attach and then enter through a hole. Using hydrolytic enzymes, they are able to break down the host cell and elongate. So when the host cell nutrients are exhausted, they form septates for the progeny Bdellovibrios. In one cell cycle, 1 to 3 hours, they are able to produce 3 to 6 progeny from a single E. coli. Usually, Bdellovibrio is grown in co-culture with E. coli in an overlay nutrient agar at 30 degrees Celsius for 1 week. So in terms of application, there are plans to use this Bdellovibrio to eradicate pathogens, for example Salmonella enteritis, in eggs. So here you can see the free-swimming Bdellovibrio in attack phase, and upon attachment, they will enter the periplasm and enter the cell. Bdelloplast is the 2-cell formation, and then as the Bdellovibrio utilizes the nutrients from the host, they elongate, and when the nutrients are near exhaustion, they will begin to septate, which means they will begin to divide into separate Bdellovibrios. Then usually when the nutrients are exhausted, the Bdelloplast will lyse and will release the Bdellovibrio, and the cycle continues. They also have a host-independent stage where they can grow independent of hosts in biofilms or in liquid as filamentous cells. So this is another diagram that shows the attack phase here, the formation of the Bdelloplast, the elongation and septate formation, and then the release of the Bdellovibrio. This is the electron micrograph showing the attachment of the Bdellovibrio onto the host cell.